Okay, next topic we are going to study about classification of virus. Now, classification of virus is a difficult task. So, here David Baltimore has tried to classify virus in the year 1971. Now, his classification is based on two aspects. First one, mechanism of RNA production, how the RNA is produced. And next is the nature of the genome. As we have studied, the genetic material in virus, it may be either DNA or RNA. Next, what is the nature of the genome? Whether this DNA or RNA, it is single stranded, represented as SS, or it is double stranded. Okay, so whether it is having single stranded RNA or double stranded RNA, or single stranded DNA or double stranded DNA. Next is the use of reverse transcriptase. Now, transcription is a process by which, as we can say, in our body, in our cells, the information is stored in the DNA. The information which is present in the DNA is in the form of a code. That code is encoded in the form of messenger RNA. This messenger RNA carries the information to the ribosomes for protein synthesis. This happens in the normal cells in our body. Whereas in the reverse transcriptase, just the reverse, the RNA is capable of synthesizing the protein. Okay. So the enzyme here involved here, it is called as reverse transcriptase. Okay. Next nature of the genome is, it is single stranded RNA. It may be positive sense or negative antisense. So what is positive sense? Now here, as we studied, this RNA it may act as a genome also. Next, it is also acting as a messenger RNA which will carry the information for protein synthesis. So here we can say the single stranded RNA, it acts as a genome as well as it acts as a messenger RNA for protein synthesis. So such type of virus is said to be positive sense. Now, antisense is what? Here the single stranded RNA will act as a genome, but it is not directly involved in protein synthesis. Okay, so here it is antisense. This is called as antisense. Next, this negative antisense first has to be converted into positive sense, then only transcription can take place. So here, based on this, the viruses are classified into 7 classes. So here, first class is virus with double stranded DNA. So example for it is adenovirus. Next, class 2 is virus with positive sense single stranded DNA. Example, parvovirus. Third class is virus with double stranded RNA. So example is reovirus. Fourth class, virus with positive sense single stranded RNA. So here example is togavirus. Next class, virus with negative antisense single stranded RNA. Example, rhabdovirus. Class 6, virus with positive sense single stranded RNA, reverse transcriptase that replicate with DNA intermediate in life cycle. So as we have studied, reverse transcriptase means, so here transcription is taking place from the reverse direction, from the RNA. So in such case, in the intermediate, DNA is synthesized. So here RNA will synthesize the DNA further will synthesize the messenger RNA. Based on that, the protein synthesis is taking place. So example for such is retrovirus. And the class 7 virus, it is double stranded DNA, reverse transcriptase that replicate with RNA intermediate in life cycle. So here it is double stranded DNA, and here the intermediate is what? The RNA is synthesized. Example is Hebna virus. Next we are going to study about the viral genome. Now each virus, they possess only one type of nucleic acid. As we have studied, virus has two components again. we are saying the nucleic acid as well as the protein code. So this nucleic acid, it may be either DNA or RNA. So this word DNA, the nucleic acid DNA or RNA is called the viral genome. The nucleic acid, it may be linear, it may be either in a straight line or circular form. Okay, so here it can be straight or it can be circular. Generally, nucleic acid is present as a single unit. So here this DNA or RNA is present as one single segment or one single factor or unit. But in wound tumor virus and in influenza virus, they are the type of virus, this nucleic acid or the genome, it is found as small segments. There are numerous fragments or pieces of genome is found in this viruses. The virus which possess the DNA, they are called as deoxyvirus, whereas those virus which possess RNA as the genome, they are called as ribovirus. So it is clear. So as we studied, nucleic acid may be DNA or RNA. If the nucleic acid is DNA, we'll call such virus as deoxyvirus. If the nucleic acid is RNA, we'll call such virus as ribovirus. The majority of the animal and bacterial virus are DNA virus. So most of the animal and bacterial virus, they have DNA virus, except HIV. HIV is a animal virus, but it possesses RNA. Similarly, plant virus, they generally contain RNA. Okay, usually plant virus, they contain RNA, but here also exception is there. Cauliflower mosaic virus, they possess the DNA. So here it is clear. Animal and bacterial virus, mostly they are DNA. Exception is HIV, which contains RNA. Plant virus, mostly they contain RNA, but cauliflower mosaic virus is an exemption. That is, DNA is the genetic material or nucleic acid there. The nucleic acids may be single-stranded or double-stranded. On the basis or nature of nucleic acid, viruses are classified into four major categories. They are virus with single-stranded DNA, example parvovirus; double-stranded DNA, bacteriophage; single-stranded RNA, tobacco mosaic virus; and double-stranded RNA, wound tumor virus. Next, we are going to study about the tobacco mosaic virus. Tobacco mosaic virus was discovered in 1892 by Dimitri Ivanskov from the tobacco plant. Virus infect healthy plants through vectors like aphids or locusts. So here they are the leaf-eating insects. They are the carrier of this virus. The first visible symptom of tobacco mosaic virus is discoloration of leaf. So a plant is affected by tobacco mosaic. The first symptom is what? Discoloration. The leaf will start losing the green color along the veins and show typical yellow and green mottling. Mottling means dying, which is the mosaic symptom. The downward curling and distortion of the young apical leaves occurs. So here the leaves, they start curling and the young leaves, they start dying. Plant becomes stunted and yield is affected. Now here, when the plant is affected by the virus, so the growth of the plant stops. So the plant shows the stunted growth and the yield is also up. Let's study about the structure of tobacco mosaic virus. Now electron microscope has revealed that tobacco mosaic virus is a rod shape. It's a rod shape virus which is helical virus measuring about 280 into 150 μm. So here it is the tobacco mosaic virus is a rod shape. In appearance it is rod shape and it is a helical. It's showing helical symmetry. The size is 280 into 150 μm and the molecular weight is found to be 39 into 10 raised to the power of 6 Daltons. The virion, that is the virus, is made up of two constituents, a protein core called capsid and a core called the nucleic acid. So here it has two components, the capsid and the RNA core. The protein coat is made up of approximately 2130 identical protein subunits called the capsomeres. So here you can see this yellow petal shaped structures. they are called as, each petal shaped structures, they are called as capsomeres. So they are the small subunits. Capsomeres, they are the small subunits, and the number of these small subunits, it is 2130, and they are all identical. So this capsid or the protein coat is made up of 2130 identical protein subunits. They are called the capsomeres. In the core, there is a single stranded RNA. So here the nucleic acid is a single stranded RNA. The genetic information necessary for the formation of a complete TMV particle is contained in its RNA. Okay, so this RNA, it contains all the genetic information of this tobacco mosaic virus, and this the number of nucleotides present in this RNA, it is 6500. So as we have studied, tobacco mosaic virus is a rod-shaped virus with helical symmetry. It has two components, the capsid and the RNA. Capsid, it is made up of 2130 identical protein subunits called the capsomeres, and it has having the core of single stranded RNA. This RNA is made up of 6500 nucleotides. Next, we are going to study about the structure of bacteriophage. Now, as we know, what is a bacteriophage? Virus infecting bacteria, they are called as the bacteriophage. So here it literally means eaters of bacteria. Okay, bacteriophage means eaters of bacteria, whereas the phage means to eat. The phages, they are abundant. These bacteriophages, they are present in soil, sewage water, fruits, vegetables and milk. The structure of bacteriophage, if we can see, it is T4 phage is tadpole shape and consists of a head, a collar, tail, base plate and fibers. So here you can see a hexagonal head is present there. At the base of the head, there is a collar is present. Below the collar, there is a tail is present. The tail is surrounded by the sheet, and at the base of the tail is present the basal plate. From the basal plate arises the fibers. The head is hexagonal. Okay, it has 6 faces, hexagonal, which consist of about 200 identical protein subunits. So this head of this bacteriophage consists of 2000 identical protein subunits. The long helical tail consists of inner tubular core, which is connected to the head by a collar. Okay, so here the tail is a core region which is connected to the head by a collar. There is a base plate attached at the end of the tail. At the end of the tail, you can see the base plate is present there. Okay, the base plate, they contain 6 spikes and tail fibers. So here from this base plate arises the tail fibers. These tail fibers are useful in attachment of the bacteriophage to the cell wall of the bacterial host. So with the help of this tail only, the bacteriophage attaches to the surface of the bacterial host during replication, during its multiplication. Now in the head, the double-stranded DNA molecule of about 50 μm is present inside. Inside the head, the genetic material double-stranded DNA is present, which is the size of 50 μm is present inside the head. The DNA is about 1000 times longer than the phage itself. So here the double-stranded DNA which is present inside the head, its size is 1000 times larger than the size of the bacteriophage. So with this, children, we complete today's class. The next class we will be studying about the life cycle of phages and the diseases caused by the virus. Thank you, children.